Hello everyone, welcome to part two. This is part two of the first chapter, this introductory chapter to microbiology. Again, this lecture and this discussion is designed for my introductory microbiology course for college students. But of course, anybody can watch this and hopefully learn something from it. So let's begin. We'll start with some history with this second part of chapter one. So we'll look at the first observations here of microbes, and we go way back to 1665. And the English scientist, philosopher, thinker Robert Hooke did many things. He reported that living things were composed of little boxes or cells, and he used the word cell to describe them. It wasn't a new term, it wasn't a new word that he had invented. The term cell had been used um, to describe, or it was used to name the rooms that monks resided in. And so they stayed in these cells, and you think of like cells in a jail, cells in a monastery, and so they reminded Hooke of what he saw in a monastery, the rooms that monks resided in, and so then he called them cells. Now, why did he think of that? Because the cells that he was looking at were cork cells, the cork cambium from a tree, like right underneath the bark of a tree. He took a thin layer of it, looked at it under the microscope, and he saw many regular patterns. Not exactly rectangular, but overall they were generally rectangularly shaped, and so that reminded him of the cells. Now, at the time he had no idea what they were made of, what their purpose or function was, but it opened up the microscopic world to show us that living things are made of smaller parts, parts that we can't see. And that was very hard for people to accept in the 17th century. But he made these observations, and he knew that he couldn't just spread this information out via word of mouth. So he drew everything, and he drew very detailed pictures, and he put it into a book called Micrographia. And in it, there's the first drawing that he saw when he saw cells. But he drew many things, hundreds of specimens. And then he shared his findings, and so he could verify that with others as well. So he opened up the microscopic world in 1665. But the one who really opened it up and looked at an incredible variety of things was Antony van Leeuwenhoek. From 1673 to 1723, he made thousands of microscopic observations, and he too drew everything and made detailed notes and sketches of what he saw. He was the first to describe live microbes. So he saw living things swimming around in, say, a drop of pond water or saliva or fecal matter. That's very appetizing. But he saw many of these organisms swimming around, darting back and forth. And the picture on the right is a replica of one of the microscopes that he made. We think he made around 250 of these microscopes, somewhere in that ballpark, and he made them himself, and he also made the lenses as well. And his microscopes were all single lens or simple microscopes. They were not compound, but they could magnify up to nearly 300x, which was incredible magnification to get out of a single lens. So he made them spherical, and he polished them, and he put them into these brass plates. And then you can see in that replica there were a couple of screws that you could turn to move the specimen up or down or left or right. And you would hold it up to your face. It was only something that was probably less than 4 inches from top to bottom. And it was a very small instrument to use. But through that lens, he saw the living microbes for the first time. He was the first person on Earth to ever see them. And he called the microbes animalcules, which means little animals, because he thought that's what they look like. Their shapes kind of reminded him of animals. And so he called them animalcules, and that name stuck for a couple hundred years nearly, almost 2 centuries. I mean, Pasteur, who we'll talk about later, in the mid 1800s and late 1800s, he even used the word animalcules. So this term stuck from the 1600s when Leeuwenhoek was using it. We'll talk more about him later when we talk about the cell. So there was this idea now that had been floating around, had been around for millennia, ever since ancient Greek philosophers, Aristotle talked about it, called spontaneous generation, that living things arise from non-living matter. And that was spontaneous generation. In other words, life just spontaneously generated. And in order to generate life, there had to be a vital force present. If the vital force wasn't present, there was no life. So this vital force was an invisible force that inhabited living things. If it was alive, it had the vital force. If it's not alive, like dirt, it doesn't have the vital force. So the vital force could enter into non-living things and bring life into them, and then you would now have a living thing from that. So there were all kinds of ideas about how this worked, and it varied from one culture to another, but they all basically had this idea that everything just spontaneously generated through the mechanism and the workings of this mysterious vital force. The alternative hypothesis is called biogenesis, where living things come from other living things. And in 1858, a German scientist, he was a pathologist, Rudolf Virchow, he said the cells arise from pre-existing cells, which would have been a revolutionary statement to make then. He was foundational in making what's called the cell theory. This was one part of it, that all cells arise from pre-existing cells. So he was definitely in favor of biogenesis and not spontaneous generation. Um, so from the cell theory, we then expanded that to say all animals are made of cells, all plants are made of cells. If it's a living thing, it's made of cells in some way, and cells give rise to pre-existing cells, and cells are the basis of all life, the basic building blocks of all living things, which was an unheard of concept in the mid-19th century. So we'll go back even further, and these are experiments related to biogenesis or spontaneous generation, either trying to prove or disprove them. There were other experiments that had done besides ones that I'm highlighting here, but we're not going to pay attention to them. These are the ones that we want to shine the spotlight on these particular ones. So in 1668, an Italian, what we would call, I suppose, a physician, which term wasn't used really back then, Francesco Redi, he was in favor of biogenesis, not in all aspects, and these scientists, many of them were not in favor of biogenesis in every living thing or every form of life. But generally speaking, Francesco was a biogenesis supporter. And what he wanted to disprove was the spontaneous generation of maggots, which are the pre-metamorphosized form of flies. So what he did was he placed decaying meat in 9 jars, and he used 4 different kinds of meat. So he had 36 jars total. He had 9 jars for each type of meat. From what I remember, he used horse meat, goat, sheep, and beef, I think, are the 4 different types of meats he used. But whatever. He just wanted to show that it doesn't matter what kind of meat. And so you see what he's setting up here. He's setting up an experiment, and I'm going to say something about that after I just briefly review his experiment. So he had these jars, and he had, in each type of meat he had 9 jars, and of those 9 jars, 3 were sealed initially, and then he took the seal off. He sealed them with cork. He sealed them, and there were no maggots found on the meat. He had 3 jars that were open, there were maggots on the meat, and 3 jars covered with fine net, which he did later. He didn't do that initially. He then realized that he put cheesecloth over them, and there were no maggots on the meat. However, there were maggots on the cheesecloth on the top of the jar. So if you can imagine this piece of cheesecloth kind of draped over the rim of the jar, the mama flies would land there, lay their eggs, and then the maggots would hatch, I guess you could say, from the eggs. But there was no meat for them to get, so they would, I guess, die off. But, of course, it didn't matter. The flies don't know if it's cheesecloth or rotting meat. They just go towards the smell, towards the scent. And this is what Francesco found. So why did he do this? Well, this is macroscopically. So the world of microbes hadn't been opened up yet. It's just about ready to be opened up with Van Leeuwenhoek a few years after Francesco here. Van Leeuwenhoek started in 1673 with his observations. So we didn't really know anything about the microscopic world at all. So Francesco is working with macroscopic organisms like the fly, and so he just simply thought, okay, I can't imagine that flies spontaneously generate, so I suppose all he did was just one day watch. He saw flies lay eggs, he saw the development of the egg into the maggot, he saw the maggot then go through the pupal stage, and then he saw the adult fly emerge. And he's like, no spawn gen here. Flies come from flies. And so then he set up this experiment, and that's what's important about this. I think it's very important that he disproved the spawn gen of flies, but at the same time, and I think this is as equally important, this was the first experiment that we know of that we have recorded with details and controls. So he didn't know it at the time, but he set up a controlled experiment. Um, where he had 3 jars that were open. He didn't mess with them. And so those would have been the control group, the 3 jars that were open to the environment. And then he added a manipulative variable or an independent variable where he sealed them with cork or he used cheesecloth. So he's manipulating these are the independent variables that he introduced. And I don't think he knew that, but it's an experiment now. And so this is the first one we have on record. So where did the maggots come from? Spawn gen supporters said from the meat. Reddy would have said from flies, you idiotas. And of course he was Italian, and so that's why he would call them idiotas. So this was the first experiment that delved into the idea of disproving spawn gen. And it was successful, and it worked. But it didn't convince everyone because spawn gen had been around for thousands of years. So this is the idea that was in the psyche of everyone. It's spawn gen. It has to be. What other alternative is there? So this was a very difficult false belief phenomenon to disprove. It took almost 200 years to do it. Fast forward to 1745, nearly a century later, John Needham, he's an English priest. He was a spawn gen freak. He was totally in favor of spontaneous generation. And now we're in the mid-1700s, and so microbes are on the scene. Antony van Leeuwenhoek had done all his observations. No one could deny that there are microscopic organisms alive all around us. And so he wants to get in this whole spawn gen debate and say that microbes spontaneously generate from broth. So he put boiled nutrient broth into flasks. He took the broth, he boiled it, and in some cases he just left it in the flask, but he didn't boil it long enough. And what happened was, because he didn't boil it long enough, there was microbial growth. He also boiled broth, and for some reason he poured it into flasks that were not sterile. He just transferred it into basically what he thought were clean flasks, but microscopically they weren't. There would have been bacteria there. Then he sealed the flasks, and he got microbial growth. So where did the microbes come from? And Needham said from the broth, that the broth, with the vital force, spontaneously generated bacteria. Biogenesis supporters would have said from the contaminated flasks, you idiot. And they also would have said you didn't boil it long enough, but they didn't quite realize it at that time. It wasn't until Pasteur came along that we realized that boiling had to be for a longer period of time in order to kill bacteria and eventually to kill bacterial spores as well. An Italian scientist, Lazzaro Spallanzani, he was in favor of biogenesis, that he believed that cells came from cells, microbes came from microbes, they didn't come from broth. So what he did was he boiled the broth in a sealed flask. So he poured the broth into a flask, he sealed it up, then he boiled it, and there was no microbial growth. He said, see, I sterilized it. I killed everything that was in it, and so as a result of killing the bacteria that were initially in the broth is now sterile, and it can't grow anything. There's nothing in there that can grow. So why no microbial growth? Well, Needham said the vital force was destroyed. Spallanzani would have said he was Italian, remember, so he would say the broth was sterilized, you idiota. This was a problem that Spallanzani could not get around, the vital force. Because all that Needham had to say was, well, you trapped the vital force in the broth, you then boiled it for a long period of time, which he did. He boiled it longer than Needham. He realized that Needham didn't boil his broth long enough. So he boiled it for a longer time, sterilized the broth, and all Needham had to say was, well, you destroyed this invisible thing. And Spallanzani's ready to rip his hair out because Needham introduces the vital force into the debate, which is an invisible thing that you can't see. And so all he has to do is say, well, you just destroyed the vital force. So imagine the vital force was stuck in the broth and Spallanzani starts to boil it and the vital force is like, oh my gosh, it's getting really hot in here, let me out. And it couldn't get out. And so the vital force was destroyed. So without the vital force, the broth couldn't spontaneously generate bacteria. Spallanzani couldn't figure out the way around it. He was infuriated about this, frustrated, and he couldn't figure out and come up with an experiment that could disprove the vital force, because now this is what had to be disproven. Not spontaneous generation per se, but now the vital force had to be wiped out and destroyed. So, Louis Pasteur, a giant. He is a giant of science, of microbiology, of chemistry, of biology. He enters this debate, and there was a Academy of Sciences that offered a prize for somebody to disprove it once and for all. So what he did was he made these flasks, and he made them with a long tube, poured the broth in it, and then he heated up the glass tube warm enough that he could bend it into this swan neck that you see in the picture. He then boiled the broth in those open swan-necked flasks. No microbes grew in the broth. He then tilted the flasks, and he also broke off the necks of the flasks, and within a few hours the flasks were teeming with microbes. Now, he also let the broth sit for a long period of time. And he had to, because he knew that he had to disprove this fake vital force that everybody would have been hanging their hats on, so to speak, saying, well, you just killed the vital force, and so therefore the broth couldn't spontaneously generate bacteria. So he let it sit, and in some cases, from what I've seen and read, he let it sit for weeks. And the reason why he did that was so that nobody could argue that he did not give the vital force enough time to make its way into the swan neck, up and then down into the broth. Because if he had just boiled it and then waited a few minutes and then tilted it or broke the necks off, they would have said, well, you didn't give the vital force enough time to weasel its way through the swan neck and get into the broth to make bacteria. So he let it sit for a long period of time so that nobody could say, you didn't give the vital force enough time. He would have given it plenty of time to find its way into the broth. And that was the genius of Pasteur. He was patient, and he knew what was going to happen because of his understanding of microbes and science and how they reproduced. So, great experiment, 1859 is when it occurred. He was given recognition of it a few years later in 1862, and then he became just a landmark, renowned scientist. So, some dates. There's a golden age, there seems to be, of everything, and microbiology has a golden age, 1857 to 1914. So from that time period, we have, from 1857 to 1914, an explosion of knowledge and discoveries in microbiology. We figured out the germ theory of disease. We figured out how bacteria reproduce. We figured out that there are bacterial spores and that they require special conditions in order to kill them. We discovered the technique of autoclaving, where we have sterilizing things, steam under pressure, relationship between microbes and disease and how they cause disease. It was an explosion of knowledge, and it went up until World War I. And then, unfortunately, with World War I, that kind of put the end of that type of study and many other forms of research and study, and as the world plunged itself into the First World War. But during that time period, great time for microbiology. Pasteur was involved in this, and a number of other scientists were as well. Remember Theodore Escherich, he was involved in this time period. Robert Koch was another giant that was involved in this time period as well. So there were a number of scientists, a lot of German scientists, that were involved in this whole idea of germs and disease and stopping the spread of it. We found out that microbial growth is responsible for food spoilage. They spoil wine by converting the alcohol into acetic acid, and this is where pasteurization has come from. So they wanted to, the French wineries, vineyards, wanted to be able to send their wine across the ocean, the Atlantic, to the United States. And they didn't have a way to do it because by the time it reached North America, the wine had spoiled. So they came to Pasteur and said, is there any way that we can preserve and prolong the life of the wine so that it doesn't spoil? Well, he demonstrated that spoilage bacteria could be killed by heat that was not hot enough to evaporate the alcohol in the wine. So this application of heat for a short time is called pasteurization, and so we name this process after him in honor of Louis Pasteur, and that's when he first discovered this technique, was in wine and preventing the growth of the bacteria that spoil the wine. So now, thanks to him, all sorts of foods now have a much longer shelf life because we understand that we can apply heat to kill bacteria but not destroy the compounds in the food that we want to preserve. And that's named after him. Just a couple of dates. Agostino Bassi showed that silkworm disease was caused by a fungus, and so you think silkworm. The 19th century, silk was a highly sought after fabric to make dresses and tapestries and curtains and scarves and a number of things. And so the worms produced the silk. Well, the silkworm was being wiped out by a fungal disease, and so we call on these microbiologists to figure out, well, what's causing this and can we stop it and save the silkworms? In 1865, Pasteur discovered that another silkworm disease was caused by a protozoan this time. In the 1840s, this Hungarian physician, Ignaz Semmelweis, he advocated hand washing to prevent transmission of puerperal fever from one OB patient to another. So this puerperal fever was being transmitted because the medical students were delivering babies in this hospital, and the medical students had just come from dissecting fresh cadavers. And the doctors in those days did not deliver the babies because they thought it was beneath them to do such a what they would consider a disgusting task, a menial task. So the medical students were the ones they were the ones that were responsible for delivering children. So they would be dissecting a cadaver. A doctor would say, hey, Sally needs to deliver a baby down in room 3. So the medical student would just wipe his hands off on his apron, no gloves, nothing, and then go down and deliver a baby. And if he had to manipulate this baby in any way, he would transmit dead, decaying flesh from the cadaver into the woman's uterus, and that would lead to what's called puerperal fever, and many of these new mothers died. And Ignaz said, hey, we should wash our hands. And take care of that. And so as a result of his work, the number of cases of puerperal fever went down. But that's only part of the story. I'll tell the rest of the story later in another chapter when we talk about transmission. 1860s, Joseph Lister, he used a chemical disinfectant at surgical wound sites to prevent surgical wound infections. He looked and read Pasteur's work showing that microbes are all in the air. Pasteur did work using cotton and trapping microbes in it, and then he cultured bacteria in it, and he's like, yeah, see, bacteria in the air. Joseph Lister reads that work and then applies it clinically to prevent surgical wound site infections by using chemical disinfectants that would kill the bacteria. 1876, another giant in microbiology, Robert Koch, he provided proof that a bacterium causes anthrax and provided the experimental steps which are called Koch's postulates to prove that a specific microbe causes a specific disease. So it's not just any bacterium causing a disease of a wide array of diseases. In many cases, it's one microbe, one bacterium causing this particular disease. Now, anthrax can enter the body in different ways, but usually microbes, pathogens, have preferred portals of entry, preferred ways of getting into the body. And so their signs and symptoms are usually characteristic. They're almost always the same. And Robert Koch discovered this, and then he replicated the disease in other organisms, lab animals, basically rabbits and mice and rats. And replicated the disease in healthy individuals and showed that, see, this particular bacterium causes this particular disease. And this video, History of Germ Theory, my students I recommend that you click on that and watch that video that gives a brief history of how germ theory came about. Obviously you can't in the YouTube, but on Canvas you can access these PowerPoints and you can click on that hyperlink. So what about vaccinations? Well, in 1796, we'll talk about that when we get to viruses, more about Edward Jenner and the first vaccination. He inoculated a boy with mild cowpox viruses. The boy then was protected from the deadly smallpox. So he developed the mild illness of cowpox. Then Jenner intentionally exposed this boy named James Phipps. He intentionally exposed him to smallpox, live smallpox viruses, and little Jimmy Phipps did not get sick. He didn't get smallpox. So Jenner discovered that whatever is in the whatever it is that he extracted from the cow, which was the cowpox virus, which Jenner did not know in 1796. We didn't know anything about viruses, but he knew there was something in those cowpox lesions that would create immunity within anyone that he scraped the cowpox, what do I want to say, the cowpox pus into. So he scraped it into their arms. They got sick for a couple of weeks. He then scraped live smallpox into the same individuals, and they didn't get sick. He started with a little boy, Jimmy Phipps, who was the son of his gardener, and then it expanded from there, and he vaccinated hundreds of people. It's called vaccination from vacca or waka. In Latin, you don't say a V, you say a W, for cow. And to remind us that this is where vaccinations came from. It all started with Jenner using cowpox to protect individuals from smallpox, and we call that immunity. So that's how vaccinations began. There's a lot more to that, and I will explain it in the chapter on viruses later. Treatment with chemicals is called chemotherapy, and so we use these chemicals to treat a number of diseases, whether we use chemotherapy with cancer, obviously, and that's typically the only time we use the word chemotherapy, but treating anything with any chemical is called chemotherapy. You're taking a drug, you're taking a chemical as a therapeutic agent. So if you take aspirin, I guess you could say you're undergoing chemotherapy. They can be used to treat infectious disease using synthetic drugs or antibiotics. And so taking an antibiotic is undergoing chemotherapy. It's kind of strange to say it that way because today we only think of it in terms of cancer, but by definition, if you're taking a chemical for therapy, it's chemotherapy. Antibiotics are chemicals produced by bacteria and fungi that inhibit or kill other microbes, and so we can use them to our advantage to kill bacteria. Quinine, the first chemotherapeutic agent, was long used to treat malaria around 1630s, and in South America, we know of with the Spanish conquest and the conquistadors, they saw that the South American natives were using this compound, which was in trees that grew naturally in those environments, and they were using it to treat malaria. So it was discovered around 1630. Now, how long had these South American natives been doing this? Who knows, centuries. But they knew that there was something in the tree that gave relief to malaria. Of course, they didn't know what it was, but they knew it worked. The first synthetic drug was developed in 1910 by Paul Ehrlich. He was a German microbiologist as well, and he did tons of work in microbiology. He's really not heard of and not given enough credit. But he made the first synthetic drug that was called salvarsan to treat syphilis. And then in the 1930s, sulfonamides were synthesized. Big date, though, of antibiotics and chemotherapeutic agents would be 1928, when the Scottish scientist Alexander Fleming accidentally discovered the first antibiotic. So he was working with bacteria, and he had petri plates, and was growing bacteria, and some penicillium fungi got onto his plates. And the genius of Fleming was that he noticed what you see in this picture. Around where the colonies were growing, the bacteria were inhibited. They wouldn't grow up to it. They wouldn't grow into it. They stayed away from it. And so he observed that the fungus made what he called an antibiotic, and in this case we call it penicillin, that killed bacteria. So in 28 he discovers it. In the 1940s, penicillin was tested clinically and mass-produced on military personnel. And then after World War II, it was then disseminated to the public. And there's the birth of antibiotic therapy in the 1950s then. And it was used widely. Since then, of course, we have dozens and dozens of other types of antibiotics. We have whole classes of antibiotics. And it all started with this simple discovery in Fleming's lab in 1928. So it took a while for it to be put out and into the public. World War II, of course, interfered with that. But it was first used then to treat wounds in soldiers, both British troops and I think American troops as well. And then after the war, it was given to the public and was mass produced. Couple of terms, ology terms. Bacteriology is just the study of bacteria. Mycology is the study of fungi. So if you see that prefix MYC, it's referring to fungi. So if you're a fungal specialist, you would be a mycologist. If you're a fun guy or girl. Parasitology, you're studying protozoan parasitic worms. And virology would be the study of viruses. So just a few ologies. Immunology is the study of immunity. Vaccines and interferons are being investigated to prevent and cure viral diseases. And Rebecca Lansfield, here's a picture of her in her elder years. She worked up into her 80s working in a lab doing work in immunology. She used immunology to identify closely related species of bacteria. So she differentiated between them. What we thought were two of the same, where what we thought was a single species of bacteria, she was able to differentiate between the two of them and realize that they were two different and separate species. And she used immunology to do that. And she pioneered that in the 1930s. So great job, Rebecca. Thank you for your work in that. And why is that important? Because specific species cause specific diseases. So she was able to differentiate between them. So she was getting more specific, less general, and she was using immunology to do that. A great development, great discovery. And our last slide we'll look at for part 2 is recombinant DNA. It takes DNA, or it recombines it from two different sources. In the 1960s, Paul Berg inserted animal DNA into bacterial DNA. He spliced them together. And the bacteria then produced an animal protein that they normally would never produce on their own, which led to the large amounts of the desired protein being formed. So you have a gene that's been taken out of an animal cell and then held together by DNA ligase, which binds the ends together. Now you have a recombinant DNA molecule, which is then reinserted now back into the bacterial cell. Typically E. coli is the one that we used for this. And then every time the bacterial cell would divide, that particular protein that we wanted would be produced. So why do this? Well, remember, bacteria divide very, very rapidly. So we can get huge numbers of bacteria in a very short period of time, which means we can get a large amount of the desired protein, as opposed to waiting for these human cells to divide in the lab, which takes hours for division. It takes too long, and you don't get enough of what you're looking for. So we capitalize and utilize the bacterial reproduction time, very short reproduction time, in order to get our desired protein, like insulin, for example, which is the first one that was the first protein that had come about that we had utilized recombinant DNA technology to do that. So that's the end of part 2. I have one more part, part 3. Part 3 will be a bit shorter than parts 1 and 2. So hope you learned some good information from part 2 here in this general discussion of introductory topics of microbiology. Thanks for watching.